This project explores the effect of turmeric and Indian ginseng on beta-amyloid-induced paralysis and LRRK2 mutation locomotion deficits. Introduction. The model organism used in this project is C. elegans, and there are three different strains of C. elegans. The N2 C. elegans, which represents the wild type, CL4176 worms, which represent the Alzheimer's disease model, and the WLZ3 worms, which represent the Parkinson's disease model. With respect to the Alzheimer's model, beta-amyloid plaques are a hallmark of Alzheimer's disease. They occur when a mutated form of the amyloid precursor protein aggregates between neurons. This inhibits long-term potentiation, causes neuron death, and disrupts cell signaling. In these mutant worms, they produce high amounts of the beta-amyloid peptides, which leads to paralysis. With respect to the Parkinson's model, we explore LRRK2, which is a leucine-rich repeat kinase, which is a protein in the ROCO family of the multi-domain proteins. They are responsible for basic life functions such as cytoskeleton and vesicle dynamics. Specifically, the G2019S mutation gene increases the activity of the kinase, which leads to mitochondrial dysfunction and the presence of Lewy bodies, which is important because they are aggregations of proteins that are hallmark of Parkinson's disease. In these mutant worms, there is increased LRRK2 kinase activity due to the G2019S mutation, which causes locomotion deficits in the worms. The two herbal compounds that will serve as our treatments are turmeric and Indian ginseng. Turmeric, with its active ingredient curcumin, is widely used in Ayurveda, which is Indian herbal medicine, for its antibacterial, antiseptic, and anti-inflammatory abilities. In fact, it's been known for its ability to quell symptoms of Alzheimer's. And there's a 4.4-fold decrease in Alzheimer's disease incidence in Southeast Asia, where turmeric is used as a spice. This is proven in scientific literature. For example, when curcumin was injected into mice brains, it reduced amyloid plaque buildup and prevented further plaque deposition with respect to Alzheimer's disease. And with respect to Parkinson's disease, in Drosophila with LRRK2 mutations, curcumin reduced the kinase activity and acted as a LRRK2 inhibitor. The other herbal compound is Indian ginseng, and it's a member of the nightshade family whose root is used in Ayurveda to calm the central nervous system and treat disorders. It contains withanamides, which decreases beta-amyloid plaque buildup, and it can also protect neurons from LRRK2 mutation-related increases in kinase activity. This too is proven in scientific literature. For example, in cortical rat neurons that are treated with the beta-amyloid peptide, the Indian ginseng was able to reduce axonal and dendritic atrophy, and it also reversed beta-amyloid-induced memory loss in Alzheimer's models. And with respect to Parkinson's models, Drosophila with LRRK2 domain losses, specifically WD40 domain, showed increased lifespan after treatment with Indian ginseng. So my research question is, what will be the effects of turmeric and Indian ginseng on beta-amyloid-induced paralysis and LRRK2 mutation, G2019S mutation-induced locomotion deficits? My hypothesis is that I predict both extracts will decrease beta-amyloid-induced paralysis and positively impact LRRK2 mutation-induced locomotion deficits. There are a few assumptions that I make when undertaking this experiment. The Parkinson's G2019S mutation will negatively impact the locomotion of the worms, and any negative locomotion will be due to this mutation. Similarly, in the Alzheimer's disease model, the beta-amyloid plaques are the cause of their paralysis, and the reason for any improvements or detriments in the worms is due to the administered treatment. The materials for this experiment were the three strains of C. elegans, the wild-type strain and the two mutant strains, Indian ginseng root extract, as well as turmeric extract, and nematode growth agar, OP50 bacteria, and Petri plates to propagate the worms, and the dissecting scope was used to collect data. Methods. 2 assays were performed, a locomotion assay for the wild-type and Parkinson's worms, as well as a paralysis assay for the Alzheimer's worms. So the independent variables were the worm strain and the herbal substances. The dependent variables were the body bends for wild-type and Parkinson's worms and the percent unparalyzed for the Alzheimer's model worms. The negative controls were the wild-type worms on untreated agar. The constants were incubation temperature of the worms, which was room temperature, OP50 food source, and treatment was administered 24 hours before the first data collection. For replicates, there were 3 trials of each treatment in each worm model, and there were 5 replicates, the worms, per plate. Quantitative method of data collection was counting body bends for the wild-type and Parkinson's worms, and the percent unparalyzed for the Alzheimer's worms. Mean and standard deviation are calculated for each of the treatments for each of the worm models, and a t-test was performed between the controls and treatments of each of the models and between the wild-type control and the Parkinson's treatment groups for body bends to see restorative abilities. A closer look into the methodology. First, agar plates were poured and broth was inoculated, and after 24 hours, inoculated broth to 6 plates, 2 for each strain, so that they could propagate. And the next day, worm plates were chunked so that each strain had 2 plates of propagating worms. Then the inoculated bacteria were added to 9 plates at a time, and these would be the treatment plates for each strain. This was prepared by adding nothing to 3 plates, which would serve as a control, 4 drops of turmeric extract to 3 plates, and 4 drops of Indian ginseng extract to the other 3 plates. These were allowed to soak for at least 20 minutes. First, the wild-type strain was chunked onto these 9 plates, and after 24 and 48 hours, the number of body bends per minute was counted. Then, the preparation of the 9 treatment plates and the chunking of the worm strains was repeated for the Alzheimer's disease model and Parkinson's disease model, and the data was collected. The only difference is for the Alzheimer's disease model, instead of counting body bends, the total amount of worms that were unparalyzed were counted and put over the total number of worms on the plate. Then statistical analysis was performed on all of the data. This slide just shows a quick visual of how the worms were seen under the microscope. For the wild type and Parkinson's worms, you can see the worm moving from side to side and bending its body, and the amount of times it did this and moved forward was counted for the wild type and Parkinson's worms. For the results, the first graph is the average body bends per minute for the wild type C. elegans. There are multiple things you can see from this graph. First of all, you can see that all the treatment groups showed an increase when compared to the respective controls. In fact, all 4 treatment groups had a significant difference when compared to the controls. You can also see that the values were higher at 48 hours, showing that the treatments were more potent at 48 hours. The next graph shows the average number of body bends per minute for the Parkinson's model C. elegans, and from this you can see that when compared to the control, each of the treatment groups did have higher values. However, 3 of them instead of all 4 had significant differences, so the only one without a significant difference was the 48 hours Indian ginseng model, and while this was greater than the control at 48 hours, it was not enough to be significant. You can also see that the values are less at 48 hours, again showing that here the potency was best at 24 hours and not at 48 hours. For the next graph, we have the wild-type control compared to the 2 Parkinson's treatment groups at 24 hours. 24 hours was chosen because there was a higher potency of the treatment at 24 hours, and we can see that both the treatment groups are greater than the control, showing that the Indian ginseng was able to not only restore locomotion to the wild-type's level, it was actually able to improve it even more than that. In fact, the Indian ginseng treatment group actually had a significant difference when compared to the wild-type control group for the number of body bends. And the last graph shows the average percent unparalysis for the Alzheimer's model C. elegans. And here again, we can see that all the treatment groups showed an increase in the average unparalysis percentage when compared to the control. In fact, all 4 treatment groups showed significant statistical difference when compared to the control. Also, you can see that the 48-hour values are less than the 24-hour values, showing that the treatment was most potent at 24 hours. So for my conclusion, my hypothesis was supported. All of the comparisons between the treatment groups and the controls had significant differences, except the 48 hours Indian ginseng and the Parkinson's model. And even that, it was a greater number, just not enough to be significant. The comparison between the wild-type control and the Parkinson's treatment group showed that not only were the treatments able to restore locomotion, they were able to improve it from the wild-type control. And the data shows the potency of these substances in relation to the characteristics of Alzheimer's and Parkinson's disease that are displayed in the differing worm models. Error and extensions. As discussed in the graphs, there were high standard deviations, and this could be due to worm-to-worm differences in body bends. As I was collecting data, you could tell that different worms moved at different rates, no matter what plate they were on, and this can be due to age or other activities like foraging. For example, if a worm stopped for way too long and did not exhibit any sign of body bends, then the calculations had to be restarted and the timer had to be restarted because that was not an effective data point. However, with increased experimentation and trials, this standard deviation can definitely go down. There was also an issue with initial contamination and temperature upshift issues when we got the stock plates for the Alzheimer's worm models, and this led to an inconsistency in the number of worms during paralysis assay on each of the plates due to the stock worms that were delivered to us being contaminated, and the temperature upshift issues affected their reproduction. There are various extensions for this project. For example, I would like to test different concentrations of the substances, test concentrated curcumin instead of turmeric to see if that would have a more potent effect, pinpoint the specific mechanisms of action of the active ingredients in the treatments. For example, they were both roots. This could have something to do with it. Also, the end game is to test efficiency in human neuron models because that is the goal of projects such as these, to find better and innovative treatments that could possibly work against Alzheimer's and Parkinson's disease.